Hi, I'm Ang Jiangming, metric number 206504. Now, I will show the way to isolate single colony using strip flat method. For strip flat method, it is divided into two parts, which are four-way strip flat technique and alternative strip flat technique. Actually, these two techniques are similar. The only difference is the pattern of drawing of quadrant on the petri dish. Before start my experiment, I will introduce my apparatus and material. The first thing is Bunsen burner. Since I'm not in the lab, so I will use lighter to represent my Bunsen burner, inoculating loop represented by the marker pen, agar plate represented by the cover of container, and lastly sample mixed culture of Serratia macensis and Staphylococcus aureus represented by this bottle with water. My experiment now. First, I imagine that my petri dish is divided into four quadrants. Since I can imagine well, so I will not draw the quadrant at the back of the petri dish. Next, follow by flaming the inoculating loop. The inoculating loop should be flamed until it turns red and let it cool down for 10 to 20 seconds. The correct way to hold the inoculating loop during flaming is like this. Do not hold the inoculating loop like this. If we hold the inoculating loop like this when flaming, we might get burned. The correct way is like this. Flaming is one of the aseptic technique. It can minimize the contamination of culture, sample culture and culture media. After letting it cool down, we proceed to our sample culture. Open the cover. Oh yeah. After flaming the inoculating loop, do not put it on the bench or any surfaces. If we put it on the bench or any surfaces, it will get contaminated again. Open the cover like this. Flame the neck of the sample culture. Dip our inoculating loop inside to obtain a loop full of sample culture. Reflame again, then close it. This is also to prevent the sample culture from getting contamination. Okay, now my loop is full of culture. I will drag it across the surface in quadrant 1. After I stop, I will use my finger to point at where I stop so that later I will not forget where I stopped. Reflame the inoculating loop again, let it cool down again, then turn our petri dish into 90 degrees. Touch the inoculating loop at the corner of quadrant 1, where we stopped at just now. Then drag it across the surface in quadrant 2. Please remember that the inoculating loop should not enter the first quadrant anymore. After that, I will point it here, turn it 90 degrees, reflame the loop, let it cool down. Again, touch the corner of quadrant 2, drag it across the surface in quadrant 3, then I point it here. Without reflaming the loop, touch the corner of quadrant 3 again, then drag it into quadrant 4. After finishing dragging the culture, reflame the loop again. The petri dish is covered with the cover of petri dish and incubate in an inverted position at room temperature for 48 to 72 hours. The reason why we need to position the petri dish in an inverted position is because to prevent the condensation from on the surface of the cover during solidification from dropping on the colonies and contaminating them directly. This also can prevent agar from drying, colonies from sinking into medium, and prevent the bacteria from overheating. Thank you.